Yes, welcome to Kenya Science Network, where we are going to network, interact. This is Biology Form 4. I am Teacher Zamalek. Welcome to the session where we are only going to talk about genetics. We are going to talk about genetics as part of the opener Term 3 examination. Stay tuned. Now, let's dive to question 1A. What is meant by the term genetics? What is meant by the term what? Genetics. Now, genetics in particular refers to, from the word genetics, refers to the branch of science that deals with heredity. Let me just enlarge my board. Deals with heredity and variation. It deals with heredity and variation of organisms. Now, we realize that there is a section of science that particularly deals with how organisms acquire characteristics. Are you getting that? So what is acquisition of characteristics? That is what we call heredity. Heredity means acquiring characteristics from parents. Are you getting that? So offspring acquire characteristics from where? From parents. Are you getting that? So that is what we call heredity. How about variation? These are observable differences. Observable differences among organisms. Are you getting that? So that is the meaning of genetics. So we are going to study inheritance or heredity, and we're going to also study what? Variations of organisms. How organisms vary. Are you getting that? They can vary in terms of height. They can vary in terms of sex. You as an individual, I as an individual, I am a male. That is my sex. You, I don't know whether you are a female or a male. Are you getting that? So how do we vary? Are you getting that? So we have what is called continuous variation and discontinuous variation. Are you getting that? So those are types of variations in genetics. So in genetics, generally, we're talking about how offsprings are being produced and how these offsprings acquire characteristics or features from their mothers, from their parents. Are you getting that? From the father and the mother. If, for instance, the child is colorblind, it therefore means that the genes, genotype, from the word genetics, we get genes, and there is what we call genotype. And there is also what we call phenotype. Phenotype is the physical characteristic of an organism. Are you getting that? What we can observe, what we can see, that is what we call physical. But genotype, these are genetic constitution of an organism. Genetic constitution therefore means that these organisms have some genes. Are you getting that? The genes are found where? Within the nucleus of any cell. And that is why we have ovum cell and sperm cell. These cells have got nucleus. And within the nucleus, we're going to get DNA. And in this DNA is where we're going to get genes. Are you getting that? And within these genes is where we have the acquired characteristics, where we are going to get the characteristics of an organism. And that is why at the end of the day, your father is brown or your mother is chocolate or dark in whichever the color. That is found where? Within the genes. They're found where? Within the genes. And where are these genes found? They are found within the DNA, within the nucleus, within the cells of an individual. And we are going to learn this in broad details. And what makes us learn is the genetics. Are you getting that? So when a child, for instance, is colorblind, are you getting that? We are able to identify where the color blindness came from. Did it come from the parent, the mother, or the father? So we are going to study heredity, acquisition of characteristics, and how these characteristics vary among different individuals. Are you getting that? So that is what we call genetics. So when we have individuals, let's say the mother and the father have given birth to 4 sons or 4 children, and these 4 children, one of them is colorblind. How can we identify how colorblindness came about? Through the study of genetics. So that is all about genetics. So whenever you ask the meaning of genetics, we have to say it is the branch of science that deals with heredity and variation among living organisms or of organisms. Are you getting that? So we are going to deal with acquisition of characteristics or features and how these features now differ in terms of one individual to the other individual. I hope you have understood the meaning of genetics. And if not, just comment down there, teacher, elaborate more on the meaning of what? Genetics. Let's dive to question B. State 2 examples of discontinuous variation. So we have 2 types of variations, just like I said in genetics. These variations among organisms, they are of 2 types. One of them is where we have different forms. There is distinct feature that is not found from another individual. That is what we call discontinuous. But continuous, we have continuous range of variation. We cannot say that this person is short or we cannot say he is tall, but there are some gaps in between. I mean, there are some ranges in between that can easily be known. For instance, we can only say that in terms of height, somebody can be 155, another person can be 156, another person can be 157. So this one is continuous. How about discontinuous? Now, let's say you are a male and the other person is a female. We don't have male, 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 female. I mean, male, 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 male. We are just having both male and female. So that means it is not continuous. It is discontinuous. We have only one form. There's only distinct feature that is found among organisms. That is what we call discontinuous variation. Ability to roll the tongue. There are those people that are able to roll their tongue. Can you do that at home? Practice it wherever you are. Can you try to roll your tongue? If you're able to roll your tongue, that is a feature, that is a characteristic that you yourself, you can do it. But there are others that cannot roll their tongues. That is what we call discontinuous variation. So those are some of the examples of discontinuous variations that we have. So I have said that in discontinuous variation, it involves a distinct feature, or there is a form of variation where there is a distinct feature of only one form. Are you getting that? So in that case, we are going to have, I have talked about some 2 examples, which I'm going to mention them down here. One, I have said ability to roll the tongue. Two, I have said sex. Sex is another example of discontinuous variation. If you want to learn more about continuous variation, just comment down there. I will take you through that. Remember, Kenya Science Network is here for if you want online tuition, we are always available. If you want trial lessons with you, we are also available. And if you want even topical revisions, we are here for you. That is why I am here for your sake. So just contact the number up here. The number is 0791937308. C. Differentiate between phenotype and genotype as used in genetics. That is simple. We have talked about it and we have said phenotype is the observable or physical characteristics the observable or physical characteristics of an individual or organism. Are you getting that? So the observable characteristic, that is what we call a phenotype. So you can easily see organism. Remember spellings. Let me just clean it so that you see it clearly, because spellings are part of biology or organisms of an individual or organisms. So that is what we are referring to as phenotype. Phenotype is the physical characteristics, physical appearance of an individual. Are you getting that? So the physical appearance or physical characteristics of an individual is what we call phenotype. In fact, I should say is the observable or physical appearance or characteristic of an individual or an organism. Are you getting that? So we can easily see, e.g., let me give example, e.g., we can say, what can we say? Let's talk about color. Let's talk about maybe a black, or a green flower. Let's talk about a yellow flower. Yellow flower, yes. Yellow flower, that is what we can see from a flower. The physical appearance, the physical characteristic of that particular flower is here, yellow. That is what we are referring to as phenotype. Are you getting that? So if we talk about yellow flower, let's say the gene for yellow flower is H. Are you getting that? So when it is this way, this is what we call genotype. And when it is this way, this is also referred to as genotype. So what is genotype? So because it was difference, just because of space, let me clear my board. Yes. So because it was a difference, I should say, e.g., yellow flower as an example, while genotype is the genetic constitution, of an organism. The genetic constitution of an organism. As simple as that. Genetic constitution, what it has, what are the genes? The genetic constitution, what are the genes? E.g., in this case of a yellow flower, we can talk about this one or this one. Are you getting that? Maybe it is normal flower, normal yellow flower, and this can be carrier yellow flower. Are you getting that? Or colorless flower. So that is what we call genotype. That is what we call genotype. The genetic makeup, genetic constitution of an organism. Are you getting that? So those are the two differences. Number two, state three structural differences between DNA and RNA. So we know that DNA is deoxyribose sugar, while RNA is a ribose sugar. Are you getting that? DNA has thymine as the nitrogenous base, while RNA has uracil, U, thymine is T, as the replaceable to thymine base. Are you getting that? So those are some of the differences we have. Now, I want you to give you an assignment. An assignment. I'm very happy with how some of you commented down there when I give out assignments. I want you to give me the third. Give the third difference between DNA and RNA. Talk about the strand. Are you getting that? Talk about the strand. Now, those are some of the differences between DNA and RNA. Structural means what it has. So we have talked about the sugar and we have talked about the bases, nitrogenous bases. I want to talk about pairing and the strand. That is the third assignment, an assignment for you. Comment down there the right answer and we will discuss about that, just like we have been discussing. All right, number 3, how is sex determined in man? How is sex determined in man? So we know that it is man that determines the sex of an individual, including women, but now men plays a greater role. Now, sex is determined through the sex chromosome. Through the sex chromosomes. Are you getting that? Through what? The sex chromosomes. That is a one mark. Sex is determined through the sex chromosomes. So thread-like structures are what we are referring to as chromosomes. Now, let's continue. The female have X chromosomes produced from the ova. Are you getting that? Male have 2 chromosomes produced from sperm. That is, so instead of saying EG, I should say that is X. That is X and Y. So bearing sperm. Y bearing sperm and X bearing sperm. So there is X and Y bearing sperms. So that is what takes place. So what happens? How is sex being determined? Now, this is what happens. We're going to have the female and we're going to have the male. And then we are going to have zygote. Are you getting that? We're going to have what? The zygote. And we're going to have lastly, sex. So we are still determining how sex is determined in man. Now, we have said that it is determined through sex chromosomes. And we have said female have X chromosome produced from the ova. So the ova only bears only X chromosome. But the sperm bears both X and Y. So what happens is that when female produces X here, we're going to have the X from the male. The zygote formed here is XX. And when there is XX, the sex now becomes female. Are you seeing the way we are determining? That is the first one. Remember, the sperm bears 2. When female bears X and the sperm bears Y, the zygote formed is going to be XY, and the sex in this case is going to be the male. Wow, we have determined sex through the man. So it is this man, the male here, the man, the male here, that is going to determine the sex of an individual at the end of the day. So even though women have only X that is in ova, the male sperm bears both X and Y and Y. So that is how they crosses, and then later on formed a fertilized egg, the zygote. And later on the child is born, can either be female or, I mean, even not being born, it can be determined. After immediately after fertilization has taken place, you can see that a zygote has been formed. And for a zygote to be formed, it therefore means that fertilization has happened. And where does it occur? Within the oviduct or fallopian tube. So what happens is that the female produces ova. And within the ova, we are only going to have what? X chromosomes. And these X chromosomes, just like I talk about them, they are found where? Within the nucleus and within the DNA is where we find the chromosomes. So these chromosomes, they are found within the genes. So genes bears the chromosomes. And now these chromosomes now, the X, Y chromosomes. Female have X. Male have XY. So what happens is that within the sperm, we are having two chromosomes, the Y and the X, that is being found within the sperm, and the female have X. So what happens is when they crosses, the female and male, the first bearing sperm has X, and that X crosses with the female X, it forms XX, which is the zygote, the fertilized one, making the sex to be a female. When the Y-bearing sperm fuses with the ovum that has X, the product is XY, which is the zygote, and now forms the male. So we have the female and the male. These are the sex. These are the sex. And that is how sex is being determined by a man. Again, if you have any problem, kindly contact me for more explanation. Number 4, color blindness is a sex-linked trait controlled by a recessive gene B. So let normal vision be what? Let's continue. And we have colorblind also there. So if a mother is a carrier and the father is normal, so we need to underline mother being a carrier and the father being normal. And remember, this is color blindness, and it is a sex-linked trait. It is controlled by a recessive gene B. So let's say, let normal vision be XB, and that of color blindness B capital, and that of color blindness be X small b. So we have those ones. Now, let's continue. What is the chance that their son will be colorblind? Show you are working. 5 marks. Wow, amazing. Let me repeat the question again. Color blindness is a sex-linked trait controlled by a recessive gene B. Now it therefore means that the gene that contains color blindness, we have given it X, X, that is B there, and that of normal vision, capital B, small b, capital B. If the mother is a carrier, so let's give them. Now let's talk about parents. I'm going to use a cross diagram. So parents, we are going to have the male, the mother, and we're going to have the father. Yes. Now, parental phenotype. Parental phenotype. What is the phenotype? I talk about the observable physical appearance. Now, the parent, in this case, the parental phenotype, mother. Mother is a carrier. And when it is a carrier, so gametes. When it is a carrier, we are going to have X capital and X small. This is for carrier. Yes, and normal father. So when the mother is normal, the father is normal, we're going to have XY. Remember, mother must have, so this is carrier mother, XX, just like I said, identifying the female sex. We are having sex XX. For the male, XY. So XX, XY. So the father is normal. If the father is normal, we're going to have that way. Are you getting that? And because it is a trait that is controlled by a recessive gene, X. Now let's continue. If a mother is a carrier and the father is normal, so this is a normal father. Normal father. And that one is a carrier mother. So parental phenotype, we have talked about mother, carrier mother. That is what we can see. Father is normal father. Normal. So in fact, instead of saying normal father, let's say normal vision. Normal vision. Father is normal vision. Mother is a carrier. So let's just give it carrier. Next step, let's move to parental genotype. The genetic constitution of the parents. Now we have said carrier mother is X capital and X small. This one is a carrier. Normal father, X capital, Y. So that is the parental genotype. Remember, there is crossing in between. So when this crosses, we are going to have, so crossing. We're going to have F1. So what that happens, we're going to have this one crosses with that one. What do we have? X capital and X capital. So this one is a normal daughter. Normal daughter. So this is offsprings. So in this case, we're going to have phenotype. F1 phenotype. So let's continue. These ones are genotype. So these are phenotype. So let's continue. Next, crossing this one with the other one, we're going to have X small and Y true. Next, we're going to have the same this one. So instead of doing that way, let's just finish with the first one. So remember, this one has crosses with the first one. It has also to cross with this one. What do we have? The crosses are not to the standard that I want. Good. So good. So we are going to have X capital Why? This space is very small. Let me just write normal. So this one, normal, daughter. Then this one, what do we have? This is normal son. You can see it. And then next, this one and this one, still we're going to have X capital, X small. And then lastly, we're going to have X small, Y. So this one is a carrier daughter. This is normal son, carrier daughter. And lastly, we're going to have colorblind son. So that is how crossing is taking over. That is how crossing takes place. Now let the vision be, normal vision be X capital, colorblind be X small. Gametes now. The mother is carrier and the father is normal. For the carrier, we're going to have the normal vision and the colorblind carrier between the two. And then normal, we're going to have normal, just like we said, the normal vision is portrayed where? Within the X capital and then the Y because father. So when you bring them here, we're going to have the parents, the mother and the father. The phenotype, mother is a carrier, father has normal vision. Genotype, we're going to have X capital B and X small b. Normal, we're going to have X capital B, just like I arranged the gametes. So these are gametes. So when crossing is taking place, we're going to have X capital crossing over with XB capital crossing together with XB capital from the normal. So we are going to have both capital BB. So that is a normal daughter because they are all XX chromosomes. Are you getting that? And then when we come to next, we are still going to have X capital B with Y, normal son because XY. And then come to the next one, X small b, X small capital B. That one is normal daughter. I mean carrier daughter because both are X's. And then we come to the son. We're going to have X, B, and Y. And that one is now where we are going to have colorblind because we said that the gene for colorblind should be X and small b. And that is what we have at the end. So the question was asking us, what is the chance that their son will be colorblind? Show you are working. Now we are not over until it is over. Are you getting that? Now let's go. So we have genotype, F1 generation. The genotype for F1 generation, we have, they are here, X capital. Next we have Y. Next, carrier. And lastly, colorblind. So we have 1, 2, 3, 4. And because there are 4, 100 divided by 4, 100% divided by 4. What do we have? 100 over 4, 250, 225. So it therefore means that the percentage for them bearing children include the following. Now, they're going to have 25% normal daughter. They're going to bear 25% normal son. They're going to bear again 25% carrier daughter. And they're going to give birth to 25% colorblind son. So let's continue. The chance. What is the chance? So if we want to get, how many sons do we have? How many sons do they have? We have talked about normal son and colorblind son. So there are two two sons. What is the ratio? So we have 1 over 2. Because there are two sons, we are going to have 1. Chance of being colorblind is just going to be 1 over 2. Or 50%. 50 what? Percent. If we add this and this, we are going to get to half of 100% is 50%. Are you getting that? So because we are talking about the sons only, chances of them giving birth to a colorblind son is just because we have two sons. So we are going to have 1 over 2 as the ratio. Are you getting that? So the ratio will be 1 over 2. And 1 over 2 of 100% is just 50%. So this should be your answer. This should be your answer. Now, these are very, very good question under genetics. And you need to ask me a lot of questions based on this one for you to easily understand. Today, the question was under colorblindness. Are you getting that? Tomorrow, I don't know whether the question will be about which color. Black color, normal color. I don't know. So those are the questions. These are the questions I expect you to ask me down there within the comment section so that we can easily tackle them. For a candidate, you need to have more of these questions because this will not miss in your exam. It will not miss. Trust me. Don't say I'm a prophet. Number 5, name the genetic disorder in humans that is characterized by inability of blood to clot. Now, that genetic disorder in human characterized by inability of the blood to clot is called hemophilia. Are you getting that? So the person is called hemophilic. Now, hemophilia is a condition, genetic disorder, characterized by the inability of the blood to clot in the normal way. In the normal way. So the blood should clot in the normal way. Remember the blood clotting process. It must clot in the normal way. And if it fails to clot in the normal way, that condition is a genetic specific. It is genetic specific. And therefore, the condition is referred to as hemophilic, being hemophilic. And that disorder is called hemophilia. Number next, state 2 reasons why females with Turner's syndrome are infertile. Wow. Again, this one is a sex or chromosomal disorder. This one is called chromosomal disorder. Are you getting that? This one is a chromosomal disorder where we are all going to have one chromosome. We are only going to have one chromosome at the end. And even after fertilization. Before fertilization, we always have one chromosome. After fertilization, again, we're going to have one chromosome. Why is that happening? Because of Turner syndrome. So infertility in female is brought by, number 1, ovary being poorly developed, So if the ovary is poorly developed, are you getting that? Leading to production of malfunctioning ova. So they are, in fact, not even leading to production of failure, leading to failure of a functioning ova. That is what we need to write. Now, if the ovary is poorly developed, it leads to failure of the production of the functioning ova. And the normal ova should be functioning normally so that when fertilization happens, the chromosomes are being crossed over. So we are going to have leading to a failure leading to a failure of functioning over. So ovary, when it is poorly developed, it can lead to Turner syndrome, infertility. So number 2, now another area where, or another reason why females with Turner syndrome are infertile. Now, let's talk about inability to produce the female hormones, e.g. estrogen, inability to produce the female hormone, e.g. estrogen, leading to failure of menstrual function, failure of normal menstrual function. Are you getting that? So the inability or inadequate production of the female hormones, that is the estrogen, leading to failure of normal menstrual functioning or function, can also cause infertility in females. So the reason why female with Turner syndrome are infertile, number 1, it causes inability to produce the female hormone, the estrogen. Number 2, ovary is being poorly developed. In this case, the females will be infertile. So we are talking about the chromosomal disorder. It is called Turner's syndrome. It causes infertility. So that marks the end of this, some of the genetics questions. It was a very, very nice to interact with you. We will be discussing more of these questions as I get them. So don't worry. When you have some of your questions or some of the tough questions based on genetics, kindly just, you can send them via the WhatsApp. The number is up here, or you can comment down there within the comment section. I will always respond appropriately. Thank you, and bye-bye for now.